You know, when you fly over a city at night, you see, like, the grid.
Oh yeah, those glowing highways and straight lines.
Right. You look out the window, you see the dense clusters of downtown lights, the avenues, and you instantly know, uh, things live there. Things built that.
Yeah, I mean, the geometry totally gives it away. It's obviously engineered, right?
Yeah, exactly.
We recognize intention in those patterns because, well, nature rarely builds in perfect grids.
But then, you know, you look at a patch of dirt in your backyard or like a piece of rotting fruit on the ground and you just think, nature.
Right, just random chaos.
Yeah, just random biology doing its thing. You definitely don't assume there's an architect at work in a rotting apple.
But what if that rotting fruit is just as heavily engineered as the city grid you see from the airplane?
Oh, man.
Yeah, what if there's a master planner
Mm-hmm.
actively curating that space, terraforming it, just on a scale we can't easily see with the naked eye?
That concept is exactly where we are heading today. Welcome back, everyone, to the next installment of our ongoing C. elegans deep dives.
It's good to be back.
Now, since you listening are already well-versed in the fundamentals of these amazing microscopic nematodes, you know we're completely obsessed with them.
Oh, totally obsessed.
And if you're loving this ongoing worm journey as much as we are, take a second to like, follow, subscribe, comment, and share this deep dive with your fellow biology nerds. We've got a fantastic one for you today.
We really do. So today we are looking at a brand new 2026 paper by researchers Bodkin, Sankaran, and Shapira.
Right.
And it flips a very basic biological assumption completely on its head. I mean, usually we think about how an environment shapes the animal that lives inside it.
Like how the amount of food available in a forest dictates the population size of deer, or how temperature dictates survival.
He's just assumed the environment is the boss.
But the central premise of today's source is the reverse. We're looking at how massive populations of worms actively reshape and engineer the microbial world around them.
It sets a really fascinating philosophical tone. Nematodes are like one of the most abundant taxonomic groups on Earth.
Yeah, everywhere.
Yeah, everywhere. And they're absolutely crucial for global organic matter cycling. Yet, mechanically speaking, we actually know very little about how they alter microbial diversity in the wild.
Huh.
This paper reveals it's a bidirectional relationship. Worms don't just passively graze on bacteria like cows in a field. They actually curate their environment.
Okay, let's unpack this. Because to understand how worms change the soil, we first need to understand the experimental world the researchers built for them.
Right, the setup is key here.
They didn't just use, sterile, boring laboratory agar plates seeded with one single type of bacteria. They wanted to emulate natural decomposing habitats.
Well, yeah, if you want to see how an animal behaves in nature, you have to give it a slice of nature. The ecosystem is part of the organism's behavior.
Literally a slice in this case.
Yes.
The researchers used actual 3-millimeter thick apple slices.
Which is such a cool methodology.
It is. They placed these on agar plates and then seeded them with 3 distinct compost microbial extracts, or CMEs for short.
Right.
So they had an apple-enriched compost, a banana-enriched compost, and a pumpkin-enriched compost. ACME, BCME, and PCME.
I love the acronyms.
Always. So you have these 3 wildly different microbial communities thriving on their own little 3-millimeter apple islands.
And the timeline of what happens next is critical.
Right. Break that down for us.
So after letting those distinct bacterial communities acclimate and grow for a couple of days, the researchers dropped exactly 30 migrating dauer larvae onto these plates.
Just 30.
Just 30. And for those tracking the life cycle, dauers are that tough, stress-resistant, non-feeding stage that C. elegans enters when conditions are harsh.
Right. They shut down their digestive systems and thicken their outer cuticles.
Exactly. They are nature's pioneers, hitching rides on insects or simply waiting it out to find new resources.
So, day 0, 30 tiny dauer pioneers touch down on the apple slice. By day 3, these dauers have sensed the environment and realized, hey, there is a massive buffet waiting for us.
Time to wake up.
Yeah. They wake up, mature into adults, and start reproducing. And this triggers the beginning of the boom.
And the population expansion is just staggering. From day 3 to day 9, the reproduction is explosive.
I mean, they breed so fast.
They really do. By day 9, the population hits its absolute peak. We're talking massive amounts of larvae swarming this tiny piece of fruit.
Oh, wow.
The environment becomes incredibly crowded. And this is also when the first worm corpses begin to appear in the soil.
Because, resources on a 3 millimeter apple slice are finite.
Very finite.
So from day 15 to day 22, the nutrients from the apple and the bacteria are essentially depleted. The population crashes.
Right, the bust phase.
The worms realize the party is over, and the new larvae arrest their development. They revert back into that stress-resistant dauer stage to wait for the next apple to fall. In total, this whole boom-to-bust cycle takes about 4 generations.
It's a classic ecological trajectory. A rapid rise, an overshoot of the carrying capacity, and a subsequent collapse, all captured perfectly in a petri dish.
Wait, here is where my brain got completely stuck reading this.
Oh, yeah.
Yeah, I'm imagining like a sudden gold rush. You have a tiny pioneer town of 30 people that explodes into a massive metropolis of tens of thousands in just days.
A nematode metropolis.
Right, and then the gold is gone and it turns into a ghost town. But with thousands of hungry worms suddenly grazing on this tiny, finite apple slice, wouldn't they just wipe out all the bacteria completely?
That is the logical assumption, yes.
How does anything survive that level of catastrophic locust-like consumption?
You would expect the worms to just strip mine the environment until nothing is left but barren dirt. But the data reveals something entirely different.
Really?
Yeah, they don't destroy the microbial ecosystem. They forcefully steer its evolution.
Huh. If you're listening and trying to map this out, it sounds contradictory at first, because the researchers measured alpha diversity and beta diversity, and the results seem like a paradox.
Right. It's a bit counterintuitive.
Think of alpha diversity as the sheer number of different bacterial species at a single party, just a raw head count of variety.
Like how many different kinds of people showed up.
Exactly. In the control plates, the ones with no worms at all, microbial diversity increased over time as the compost decayed. But in the worm-grazed plates, that alpha diversity also increased.
Yeah.
The worms didn't eliminate the variety of species.
Right. The raw number of different species, the alpha diversity went up regardless of whether worms were there or not. But beta diversity is where the mechanism of their engineering hides.
Okay, what's the difference there?
Well, beta diversity measures the differences between communities. It asks, are the specific guests at this party the same as the guests at that party over there?
Gotcha.
And the beta diversity followed a totally different trajectory on the worm plates compared to the controls.
So how can you have more variety but look the same? Because this leads to the biggest aha moment in the whole paper.
The convergence.
Yes, the concept of convergence. They started with 3 highly distinct environments, right? The apple, banana, and pumpkin composts.
Completely different starter microbial communities.
Yeah, if you looked at their bacterial profiles on day 0, they looked nothing alike, different guests at different parties. But by the end of the experiment, the presence of worms caused these 3 distinct environments to become incredibly similar to one another.
What's fascinating here is the sheer force of that biological pressure.
Yeah.
To be fair to the data, transitioning to a common substrate because all the composts were eventually put onto an apple slice.
Right.
That did cause some convergence even in the control plates without worms. That's expected. The food source dictates the survivors to a degree.
Sure. Like if you host 3 different parties but you only serve pizza at all of them, eventually only the people who like pizza will stick around.
Exactly. But the researchers found that the presence of worms drove significantly more pronounced convergence than just the food alone.
Wow.
Yeah, the differences between the early environmental microbiomes and the late ones on the worm plates were stark. The worms are actively acting as ecosystem engineers.
So they aren't just eating indiscriminately.
Not at all. They are reshaping different habitats into a unified, worm-friendly microbial landscape.
They are terraforming the apple slice.
Basically, yeah.
But how? If the environments are all starting to look the same, what exactly is happening on a microscopic level? Because, to have convergence, you need clear winners and losers.
You do. And to figure out who was winning and losing, the researchers used 16S rRNA gene sequencing.
And for those wondering how we even know this, think of 16S RNA sequencing as scanning the barcodes at a grocery store checkout.
That's a great way to put it.
It's a genetic marker that lets scientists identify exactly which bacterial families are present and in what quantities. And when they scanned the barcodes of the worm-grazed lawns, the absolute biggest losers were the Pseudomonaceae family.
Mm-hmm.
In the control plates, these bacteria were thriving, but in the worm plates, their numbers plummeted.
The implication there is quite simple, actually. Worms love to eat them.
Oh, really?
Yeah, they are a highly preferred food source. As the worm population booms, they graze heavily on Pseudomonadaceae. They eat them much faster than the bacteria can reproduce, severely depleting them in the environment.
Okay, I have to push back on the evolutionary logic here.
All right, let's hear it.
If worms love them and they eat them until they practically disappear from the environment, isn't that an evolutionary failure? I
All
mean,
right.
why would an organism terraform an environment just to eliminate its preferred food source? That seems like a terrible long-term survival strategy.
It seems like a failure if you view it as a steady-state system, but remember the boom and bust cycle. The Pseudomonaceae are essentially the fast-burning kindling. The worms use this highly nutritious, preferred food source to fuel that initial explosive population spike the boom.
Oh, I see.
They overgraze it, yes.
Yeah.
But by clearing out that dominant bacterial weed, they leave ecological room for other, more sustainable bacteria to flourish.
Oh, wow. They clear the forest to plant the crops.
Exactly.
Because if Pseudomonaceae are losing, someone is winning. I looked at the list of bacteria that actually increased in the environment specifically because the worms were there.
Get ready for some long names.
Yeah, I'm going to try my best. It's families like Comamonodaceae, Sphingobacteriaceae, Flavobacteriaceae, and Xanthomonodaceae.
It's quite a mouthful of nomenclature for sure, but the specific names are less important than the role they play.
Well, here's where it gets really interesting. All of those winning bacteria, the ones flourishing in the dirt because of the worms, have something in common.
They do.
They are all known members of the C. elegans core gut microbiome.
If we connect this to the bigger picture, the profound implication is that the worms are acting as a biological reservoir.
They aren't locusts.
No.
They are farmers.
Humans didn't invent agriculture. Microscopic worms have been farming their own bacteria in rotting fruit for millions of years.
Mechanically, that is essentially what is happening.
That's incredible.
Worms eat certain bacteria from the environment. They harbor them safely inside their guts, which act almost like biological fermentation of vats.
Right.
Inside the gut, these specific bacteria are protected from outside competition. They multiply, and then the worms excrete them. They disseminate them back into the environment.
So the worm gut is acting like a nightclub bouncer.
A bouncer.
Yeah. It destroys the pseudomonads at the door, but gives VIP access to the Commomonidaceae, letting them party and multiply inside before sending them back out into the soil.
That's actually a really good analogy. They show up, set up camp, and literally start seeding the dirt with their own preferred gut flora. The environment shapes the worm's gut, but the worm's gut is actively fighting back to shape the environment.
Exactly. The enrichment of these specific gut microbiome members in the grazed environments proves the host is actively seeding its own niche.
Man.
But to fully grasp how this mechanism works, we have to look at what is happening inside the worm itself during this process. Because as the population explodes and starves, that internal fermentation vat has to be reacting.
So if they are seeding the soil with their own preferred bacteria, the inside of their gut must be heavily regulated. It can't just be a passive tube.
Right.
How does the worm's internal ecosystem handle this massive influx of food during the boom phase and the sudden loss of it during the bust?
It handles it by going through dramatic physiological shifts. The researchers were actually able to break down the timeline of the worm's internal gut microbiome into 3 distinct phases.
Okay, walk us through those internal mechanics. What is happening inside the pioneers?
So phase 1 occurs right at the start, from 2-3 to day 6. This is the transition from the pioneering dauers into second-generation adults.
Okay.
And interestingly, this phase is marked by a significant decrease in internal gut microbial diversity.
A decrease. But they just woke up on a massive buffet? You'd think their guts would be overflowing with every type of bacteria on their apple.
You would, but remember what is physically happening to the worm. The pioneers arrived essentially empty.
Right, dauers don't eat.
Dauers don't have much of a functioning gut microbiome because they aren't eating. When they wake up and start consuming, they initially take in everything. But as they mature and as the second generation is born and undergoes normal development, the anatomy of their gut changes.
How so?
They start producing specific digestive enzymes. The gut lining itself changes.
So it transitions from an open door to a highly selective filter.
Exactly.
The body starts deciding who gets to live in the gut and who gets digested for fuel. So the overall diversity drops because the worm is specializing its internal workforce.
Precisely. The gut becomes a strictly managed greenhouse. And that leads us to phase 2, which is the boom phase, spanning roughly day 6 to day 12.
Okay,
During this period, the internal microbial diversity stabilizes. The overall gut composition across all three different compost types becomes highly similar.
So even if one worm is out there eating the pumpkin compost and another worm is eating the banana compost, if you look inside their guts, they look identical.
Completely identical. This is when the true core gut microbiome is fully established.
Wow. They've finished remodeling the internal house, and they are churning out the specific bacteria they want to seed back into the soil.
The consistency is remarkable. In
Yeah.
fact, a cool side note from the paper, they actually identified Clostridiaceae as a new consistent addition to this known core list during this phase.
Oh, really?
Yeah, it highlights how robust that core microbiome is during times of plenty. The mechanical filtering of the gut is working perfectly.
But biology is rarely stable forever.
No, it isn't. Phase 3 hits around day 15 and beyond. The bust.
The ghost town phase. The kindling has burned up, the resources are depleted, and the population is collapsing. What happens to the internal fermentation vat then?
It breaks down.
Seriously?
Yeah. As starvation sets in and dowers once again take over the population, the gut diversity inside the remaining active worms absolutely crashes.
Wow.
And unlike the boom phase where everyone's gut looked identical, during the bust phase, the microbiomes of worms across different plates start to wildly diverge from one another.
It's chaos.
Pure chaos.
When times are good, the gut operates like a perfectly regulated machine selecting the same healthy bacteria. But when starvation hits, the barrier functions likely start to fail.
That's the leading theory, yeah.
It's every worm for themselves, and their microbiomes fracture. They become completely unpredictable.
It's a state of deep dysbiosis. Dysbiosis simply means an imbalance in the microbial community, where the bad or opportunistic bacteria start to outnumber the good.
It really makes me think about our own biology. Well, just like human guts respond dramatically to changes in diet and stress, the worm's internal ecosystem is deeply tied to its food scarcity.
Oh, absolutely.
Like when we get stressed or when our diet radically changes, our microbiome goes into a state of dysbiosis, just like these worms in the bust phase. Our internal barrier functions get compromised.
It's a beautifully scalable biological principle. The rules governing a microscopic nematode's gut under starvation stress are remarkably similar to the rules governing larger, more complex systems.
So to bring all these incredible threads together, the grand takeaway here is that nematodes aren't just passive grazers.
No, they aren't.
They aren't just simple recycling machines blindly eating compost until it's gone. They are active modulators of global ecosystem.
Ecosystem engineers.
Yes. Next time you look at a patch of soil in your garden or a rotting apple fallen from a tree, realize you aren't just looking at dirt or decay. You're looking at an incredibly complex, actively terraformed alien world built by microscopic architects. They are farming the soil.
This raises an important question, one that extends far beyond the petri dish and the apple slice.
Okay, lay it on us.
If microscopic worms can actively terraform their environments to perfectly match their own internal gut microbiomes over just 4 generations, what invisible microbial consequences are humans having on the environments we engineer?
Oh, wow. I hadn't even thought about the macro scale equivalent.
We build these massive grids, these cities we talked about at the beginning. We think we're just changing the macroscopic landscape with concrete and steel.
Right.
But by concentrating our populations, altering our diets, and changing the physical space, are our cities and homes just macro-scale versions of the worm-grazed apple slice?
Are we forcing a massive microbial convergence around the globe just by existing in such high densities?
Exactly.
We think of ourselves as so separate from nature, but we might just be curating the global microbial landscape exactly the same way the worms curate the compost.
We are the ultimate ecosystem engineers, and we carry our own core microbiomes with us. It stands to reason that we are seeding our urban environments, shaping the invisible world to reflect our internal state.
That is a haunting and brilliant thought. We are the worms on the apple, curating a microbial world we can't even see. I'm going to be thinking about that every time I look out an airplane window from now on. The city grid isn't just concrete, it's a massive biological fermentation vat.
Pretty wild to think about.
Well, thank you all so much for joining us on this deep dive into the invisible architecture of the world around us. Again, if you love exploring these complex biological mechanisms as much as we do, please remember to interact with the show, like, comment, subscribe, and share your thoughts.
That's what we think.
Do you feel like an ecosystem engineer today? Keep your curiosity alive, and we'll see you on the next deep dive.